Unit 4 Genetics and Molecular Biology for GAT-B 2026. As given in the slide, this is one of the most important units because it connects molecular concepts with inheritance patterns. You should not treat this as separate topics like DNA, RNA, and genetics. Instead, everything it is interconnected. In GAT-B 2026 exam, many questions are directly or indirectly coming from this unit because it forms the foundation of biotechnology. So while studying, your focus should be in the understanding the logic behind processes like replication, transcription, and inheritance rather than just memorizing definitions. Why BioBrain's preparation approach? As mentioned in the slide, the preparation approach is concept-based learning along with MCQs and previous year questions. This is very important because GAT-B is not a rote learning exam. If your concepts are clear, even difficult questions can solve easily. Also, structured preparation is required for understanding theory to practicing questions and revising regularly. The key takeaway from the slide is that consistency and conceptual clarity are more important than just reading notes once. Unit overview. It includes gene structure, chromatin organization, central dogma, gene regulation, Mendelian and non-Mendelian genetics, microbial genetics, and DNA repair mechanisms. So basically, it covers everything from DNA structure to genetic diseases. You should use the slide as a checklist. While studying, always keep track of whether you have covered each topic properly. Also, try to link topics. For example, mutation connects with DNA repair and also with genetic diseases. This kind of integration is very important for exam. Molecular structure of a gene. A gene is a functional unit of DNA that produces RNA, which can be coding or non-coding. In eukaryotes, gene structure includes promoter regions, transcription start site, UTRs, coding region ORF, and terminator. Promoter elements like TATA, CAT, and GC boxes help in binding of transcription factors. Another important point is the presence of introns and exons. Introns are removed by spliceosome and this process allows alternative splicing, which means one gene can produce multiple proteins. In prokaryotes, introns are absent and genes are organized into operons, producing polycystonic mRNA. This difference is very frequently asked in exams. Chromosome and chromatin architecture. DNA is organized into chromatin through different levels of packaging. It starts from DNA double helix, then nucleosome formation, followed by higher order structures like 30 nm fiber and loop domains, finally forming chromosomes. Nucleosomes consist of DNA wrapped around histone proteins, and histone modifications plays a important role in gene expression. For example, acetylation generally activates transcription, while methylation can either activate or repress depending on the context. Also, euchromatin is active and loosely packed, whereas heterochromatin is inactive and tightly packed. These concepts are very important for conceptual MCQs. Mutations, classification and consequences. Mutations are changes in DNA sequence and can be classified into different types. Point mutations include transition and transversion, and based on effect they can be silent, missense, or nonsense. Frameshift mutation can occur due to insertion or deletion and are usually more severe. There are also concepts like forward mutation and reversion, where mutation either occur or gets reversed. Loss of function mutations reduce protein activity, while Gain of function mutations create new or enhanced activity. Dominant negative mutations are especially important because they interfere with normal protein functions, and these are often asked in higher level questions. Mutagenesis mechanisms. Cause to effect. Mutations can arise spontaneously or due to external factors. Spontaneous mutations include shift and deamination. These are naturally processes occurring inside the cell. Inducing mutations are caused by external agencies like UV radiation, ionizing radiation. For example, UV radiation causes thymidine dimers, which distort DNA structure. Chemical mutagenesis can cause mispairing or frameshift mutation. Amis test is used to detect mutagenetic potential of substances. In exam, always relays the mutagen with the type of mutation it causes. DNA replication is semi-conservative and bidirectional. It starts at origin of replication (ORIC) in prokaryotes and multiple origins in eukaryotes. Several enzymes are involved, each with specific functions. Helicase unwinds DNA. SSP proteins stabilize single strands. Primase synthesize RNA primer. DNA polymerase III synthesize new DNA. DNA polymerase I remove primer, and ligases join fragments. Leading strand is synthesized continuously, while lagging strand is synthesized discontinuously as Osaki fragments. Sliding clamp increases efficiency of polymerase. These enzyme functions are very important according to the exam. DNA replication is highly accurate due to proofreading activity of DNA polymerase. This enzyme has 3' to 5' exonuclease activity, which means it can remove incorrectly added nucleotides immediately during replication. Because of this, the error rate becomes extremely low. In addition to proofreading, mismatched repair mechanisms further correct any remaining errors after replication. However, in eukaryotic cells, there is a limitation called the end replication problem. During replication of the lagging strand, the very end cannot be fully copied, leading to shortening of telomeres. Telomerase enzymes solve this issue by adding a repetitive sequence at chromosome ends. This enzyme is active in germ cells and cancer cells. Transcription is the process where RNA is synthesized from a DNA template. In prokaryotes, RNA polymerase along with stigma factor recognized promoter region such as -10 and -35 sequences and initiates transcription. In eukaryotes, the process is more complex and requires multiple transcription factors. For example, TFIID binds to the Tata box and TFIIH helps in unwinding DNA. Elongation occurs in 5' to 3' direction, and termination in prokaryotes can be either row dependent or row independent. Additionally, phosphorylation of the CTD C-terminal domain of RNA polymerase II regulates the transition from initiation to elongation. These differences between prokaryotic and eukaryotic transcription are very important for GAT-B. RNA processing is a crucial step that converts pre-mRNA into matured mRNA in eukaryotic cells. The first modification is the addition of a 5 cap, which is a 7-methylguanosine structure. This cap protects mRNA from degradation and also helps in initiation of translation. The second important modification is the addition of a poly A tail at 3-N, which increases mRNA stability and helps in nuclear export. Slicing is another key process where introns are removed and exons are joined together following the GUAG rule. During splicing, a lateral structure is formed. Alternative splicing allows a single gene to produce multiple proteins, isoforms, which increases protein diversity. mRNA editing is also seen in some cases where nucleotide sequences is modified after transcription. Translational is the process where mRNA is decoded a protein. Ribosomes are the main machinery involved, consisting of 70S in prokaryotes and 80S in eukaryotes. tRNA molecules carry amino acids and are changed by aminoacyl tRNA synthase is an ATP-dependent manner. Gene regulation in prokaryotes is mainly explained by the operon model. An operon consists of promoter, operator, structural genes, and a regulatory gene. These genes are transcribed together as a single polycystonic mRNA. The lacoperon is an inducible system where the repressor binds to the operon and blocks transcription in the absence of lactose. When lactose is present, it acts as an inducer, allolactose, and inactivates the repressor, allowing transcription. Advanced prokaryotic regulation, a fine-tuned mechanism seen in the trpoperon. It depends on the leader sequence, which contains multiple tryptophan codons. This mechanism links transcription and translation together. When tryptophan levels are high, ribosomes move quickly across the leader sequence, leading to formation of a terminator, hairpin, and transcription stops. When tryptophan levels are low, ribosome stalls, allowing formation of an anti-terminator structure and transcription continues. This mechanism is possibly only in prokaryotes because transcription and translation occur simultaneously. Gene regulation in eukaryotes is more complex and occurs at multiple levels. These include chromatin remodeling, transcriptional control, post-transcriptional processing, translational regulations, and post-translational modifications. Enhancers plays a major role and can act even from distant locations through DNA looping. The mediator complex connects transcription factor with RNA polymerase II. Another important concept is combinatorial control, where multiple transcription factors work together to regulate a single gene. This complexity allows precise control of gene expression in different cell types. Epigenetic regulation. It refers to the changes in gene expression without altering the DNA sequence. DNA methylation. Histone modification also plays a key role. Acetylation of histones generally activates transcription, while deacetylation represses it. Chromatin remodeling complexes helps in repositioning of nucleosomes. Other important concepts include genomic imprinting, where only one parental allele is expressed, and X chromosome inactivation, which leads to formation of barbary in females. Mendelian laws. From the basis of classical genetics, the law of segregation states that alleles separate during gamete formation, while The law of independent assortment applies to genes located on different chromosomes. The typical monohybrid ratio is 3:1 and dihybrid ratio is 9:3:3:1. A test cross is used to determine genotype by crossing with a homozygous recessive individual. There are also extensions like incomplete dominance, where phenotype shows intermediate expression, and codominance, where both alleles are expressed. Multi-alleles such as ABO blood group add further complexity. Gene interaction refers to cases where multiple genes influence a single triad. Epistasis is a key concept where one gene masks the effect of another. Different types of epistasis give modified phenotypic ratios, such as 9:3:4 for recessive epistasis and 12:3:1 for dominant epistasis. Complementary genes produce a 9:7 ratio where both genes are required for expression. Duplicate genes produce a 15:1 ratio due to redundancy. These modified ratios are very important for solving genetic problems. Complementation test is used to determine whether two mutations are in the same gene or different genes. This is done by bringing mutations together in the same cell. If the normal phenotype is resorted, it means mutations are in the different genes and they complement each other. If phenotype is not resorted, mutations are in the same gene. The concept of cis and trans configuration is also important. Complementation groups represent different functional genes. This concept is widely used in microbial genetics and gene mapping. Linkage refers to the gene located on the same chromosome that tend to be inherited together. However, during meiosis, crossing over occurs in prophase 1, which leads to recombination. Recombination frequency is calculated as the percentage of recombinant offsprings. If recombination frequency is less than 50%, it indicates linkage between genes. This concept is very important for genetic mapping and solving numerical problems in exams. Genetic mapping is based on recombination frequency, where one centimorgan corresponds to one recombination. 2-point mapping gives only one distance between genes, while 3 points mapping helps in determining both gene order and distance. However, double crossovers can affect mapping accuracy, which is why interference is considered. Interference is calculated during the coefficient of coincidence, and positive interference means fewer double crossovers than expected. This concept is very important for numerical questions in GAT-B, especially in 3-point test cross problems. Extrachromosomal inheritance refers to genetic material present outside the nucleus, mainly in mitochondria and chloroplast. These organelles contain their own DNA and replicate independently of nuclear DNA. This type of inheritance is usually maternal because the cytoplasmic of zygote comes mainly from the egg. Heteroplasmy refers to the presence of both normal and mutated mitochondrial DNA within the same cell. Diseases like mitochondrial myopathies are example. These trials does not follow Mendelian inheritance patterns, which makes them important conceptual question. Microbial genetics. Plasmids are small, circular, double-stranded DNA molecules that exist independently of the bacterial chromosome. They have their own origin of replication and can vary in copy number. Some plasmids have low copy, while other high copy depending on their replication control. Different types of plasmids include F plasmid for fertility, R plasmid for antibiotic resistance, episomes or plasmids that can integrate into the host chromosome, such as F plasmid forming HFR strains. Plasmids incompatibility group determine whether two plasmids can coexist in same cell. These concepts are important in both microbial genetics and biotechnology. Transformation is the uptake of naked DNA from the environment by a competent cell. This can occur naturally or can be induced artificially using methods like calcium chloride treatment or electroporation. Transcription involves transfer of DNA through bacteriophages. Generalized transcription transfer random DNA, while specialized transcription transfers specific genes near to prophage. Conjugation involves direct cell-to-cell contact through asexpilus, where F plus cells transfer DNA to F minus cells. HFR strains can transfer chromosomal genes. These mechanisms are very commonly asked in comparison-based questions. Horizontal gene transfer refers to the transfer of genetic material between unrelated organisms. Unlike vertical transfer from parent to offspring, that mainly occur through transformation, transduction, and conjugation. This process plays a major role in microbial evolution by allowing rapid acquisition of new traits such as antibiotic resistance. It also leads to formation of pathogenicity islands in bacteria. Genomic features like unusual GC content or codon usage patterns can indicate horizontal gene transfer. This concept is very important in understanding bacterial adaptation. Transposable elements are mobile DNA sequences that can move from one location to another within the genome. They are also known as jumping genes. These are two main types: DNA transposons, which move by cut and paste mechanism using transposase enzyme. RNA interference is a mechanism of gene silencing at the post-transcriptional level. It involves a small RNA molecule such as siRNA and miRNA. siRNA shows perfect complementarity with target mRNA and leads to its degradation, while miRNA binds partially and inhibits translational. The process involves dicer enzyme, which processes double-stranded RNA into small fragments, and RISC complex, which binds and silences the target mRNA. DNA damage can occur due to internal or external factors. Endogenous sources like reactive oxygen species and replication errors, while exogenous sources include UV radiation, ionizing radiation, and chemical agents. Different types of DNA damage includes single-strand breakers, double-strand breakers, and base modifications. UV radiation causes thymine dimers, while ionization radiation causes strand breakage. Understanding the type of damage is important because it determines the repair mechanism involved. DNA repair mechanisms. Cells have multiple repair mechanisms to maintain DNA integrity. Direct repair involves reversal of damage, such as photolase repairing thymine dimers. Base excision repair corrects small base changes, while nucleotide excision repairs removes bulky lesions. Mismatched repair fixes replication errors. Double-strand breakers are repaired by homologous recombination, which is accurate, or non-homologous end joining, which is error-prone. These pathways are very important for maintaining genome stability. Chromosomal variation can be numerical or structural. Numerical changes include aneuploidy such as trisomy 21 and polyploidy, which is common in plants. Structural variations include deletion, duplication, inversion, and translocation. These changes occur due to non-dysfunction or chromosome breakage. Such variation can lead to genetic disorders and are important for understanding chromosomal abnormalities. Genetic diseases arise due to mutation that lead to defective proteins and alteration cellular functions. This mutation can be different types, such as loss of function mutation where protein actively is reduced or absent, and gain of function mutation where the protein acquires a new or toxic function. Example include sickle cell anemia, which caused by a missense mutation in the beta globin gene, and cystic fibrosis caused by deletion of phenylalanine at position 508. Chromosomal disorders such as Down syndrome and Turner syndrome arise due to numerical abnormalities. Concepts like are important because explain variation in disease presentation among individuals. Integrated GAT-B 2026 exam focus. The slide summarizes the most important areas for exam preparation. High-yield topics include operon models and epigenetic regulation, DNA replication enzymes and repair pathways, genetic mapping numericals, and microbial gene transfer mechanism. Common mistakes include applying Mendelian ratio to link genes, confusing siRNA with miRNA, and mixing up homologous recombination with non-homologous end joining. The correct strategy is to integrate concepts with application and practice numericals regularly. This unit requires strong conceptual clarity rather than memorization. Stay connected with BioBrain for latest GAT-B 2000 preparation tips. Also, subscribe BioBrain for more content.